So first of all, I think I'm doing time-restricted feeding, and that's why we look like identical twins.
Exactly, yeah.
First of all, I really would like to thank the young people here, so many of you, and I'm so excited for you. You know, it took us decades to go from hope to promise, and now you're going to realize the promise, and you're going to change the world. And I really think that you're smart just because you chose this profession and to be here, and I'm really, really excited for you. By the way, there are older people, you know, Sami Segal sits here, he's a billionaire that invests, and he'll also will change the world. So it's not only young people, but I really wanted to start with something that I showed last year. I know not everybody was here. I'm sorry the politicians haven't been here, haven't stayed here to see that, but that's our life, right? Our life is a double-deck toilet, okay? And the management is on top and the employers are on bottom. And you could say that what he's done now, like shouting at us, moving us from the back, hitting us with a microphone, you know, is a total management act. When in fact, you know that we were on top. I mean, to undertake this kind of a program and to get this communication that we're getting is really unbelievable. It's first class. And I want to thank all of us to thank those guys now because they're falling asleep and tomorrow they won't be able to hear when we clap for them. So guys, Daniela, Morten, Alex, all the students, everybody. One challenge, you know, just as I showed that last time, I also said the same thing last time, that it was the best ever, it was a great meeting. So you have to do better next year so I can say the same things. Okay, so I think we all understood that we need those biomarkers of aging to move on. They're very important for us. And I think that they're really important for drug development, okay? And a lot of the people here are from the longevity biotech, and think of them, they have to get to phase 2 studies that will show some trends or even significance in biomarkers, so they wouldn't spend for everything the $1 billion of a phase 3 trials, right? I mean, this is very important for us to solve now. And I want kind of to depict some of the problem, but I'll start with depicting this problem with my own data. This is my Grimm age by Horvath. And I'm 3 years younger than my age. And I called Horvath and I said, I'm really disappointed. He said, why? I said, look, the Mayo Clinic has done this biological aging clock from 250,000 EKGs. Okay, it's absolutely quite amazing because they can tell you, I can read the EKG and say if you're male or female by your name, quite often. But they do it by EKG. When I ask them, what is it that it's female? They don't know, the cardiologists don't know, but AI knows that, okay? So AI has done biological age of the muscle, okay? By the way, of the heart muscle. So if your muscle is good and you get coronary the next day, okay, that's a problem. But what's the age of your muscle? And you see here the correlation. This is not working. You see the correlation of EKG with age. And I've been in the Mayo last year and 10 years before. So I have 2 EKGs. In one EKG, when I was there, I was 58 years old. and my biological age was 54. Can you see it now? Yeah, my biological age was 54. Since then, I've done two things. I started here on metformin, and I started time-restricted diet, a few years later. And last year, my biological age, I was 67, and biological age was 55. So I said to Horvath, you really owe me, 10 years. He said, well, can you explain why they're right and not I'm right? I said, yeah, I can absolutely explain it to you. Because when you get the Grimm age, you get the correlation of the epigenetic data with some biological measurement, okay? There is for example, leptin and pi1 and some things that make sense that are epigenetically modulated by aging. But as you see, you don't see it so well, but you see in red, there's one measure that is up in my hemoglobin A1C. And I told Steve, I understand why my hemoglobin A1C looks high, because my mother was diabetic. I have 4 genes for diabetes. I was pre-diabetic before I started metformin. So I can understand why this happens. But in those last 10 years, my hemoglobin A1C was 5, okay? Which means that this epigenetic test didn't measure my intervention successfully. Okay, and I think that's the challenge with epigenetics. Not that it doesn't tell you what's your biological age, but it doesn't really predict your age in all the parameters, your success in intervention in all the parameters you're doing. And I have studies where I have the proteomics and the metabolomics and the methylation of subject, but I'm most excited about proteins. And I think you got a little bit of a taste for that. Luigi is excited about protein, and he showed you some data on the spliceosome of proteins and how they react. Tony Weiss-Corey showed you, and this is studies that we had together, on how you can actually identify which proteins are coming from which organ, so that you can, maybe there's an organ that's leading your aging, and maybe you should know about it and do something about it. So I'm not going to repeat any of those, but I'll tell you some cool things that we discovered. And the base of the study is basically proteomics that we've done on 1,000 people who were between the ages 65 and 95. I'll tell you soon more about those people. And we asked, what changed between 65 and 95, okay? And you already know to look at volcano plots. This is a statistical, this is the p-values, and this is going up, this is going down, and the effect size. And you see that there are 17% of the proteins that are changing significantly. And you see in capital already that some of those proteins, from a work of Jamie Justice there, those are proteins that carefully were vetted and we're pretty sure are changing with treatment, okay? So that's good. We have some of those, and there are certainly some that are changing with aging. But then you ask, okay, what do we learn with the other proteins? Do we find the causes of aging, okay? And the answer is it's very complicated because, for example, PTN and GDF15 in animal models will live longer. So is it a protective mechanism for aging? I mean, you think, okay, those proteins are going up, let's get them down. But in fact, those proteins should go higher maybe, okay? So we were looking at that. Remember that that's very confusing. We have to make sense of that. So one way to make sense of them is to look at the proteomic pathway and understand where are they coming from. And there are 2 points I want to make here. One is that on top of our genetics in our study, in tops of our proteomics in our study, in top of our clinical thing, are always things that are related to growth hormone IGF signaling pathway. Okay, this is really a major pathway that always comes up. But those things in red are the most interesting thing. I cannot read it in this computer, but what you have is extracellular matrix degradation, platelets degranulation, collagen degradation, neutrophil degranulation, collagen biosynthesis. And at first I said, okay, you know, that's not helpful, until I realized what we're seeing here is really the breakdown of aging. This is kind of the definition of aging. We measure it in the plasma. We get lots of things that are breaking down. And maybe, no matter how we treat aging, what we need to stop eventually is the breakdown. Maybe that's the common mechanism that will break with whatever we are doing. Okay? So that's point one. Point two, Tony has shown that you can look at which organs they are coming from, but it's more than that. You can look also at specific diseases, and frailty is one of the age diseases, age diseases. And what you see here, that when we look at people who are frail versus who are not frail, we have some common things that happen with aging, but more interesting, almost half of those proteins are not part of the aging. They are specific from frailty. Okay? Look for example at leptin levels. Leptin levels are very prominent in a frailty, and maybe there's actually a physiological explanation. But the point here is that maybe we can identify frailty, maybe we can identify pre-frailty by some of those proteomics that are here. So let me tell you about this population of 1,000 people. So I have a study. Last year I presented my study on centenarians. We have centenarians, 750 centenarians. But we have a longitudinal study on the offspring of centenarians and people who are usually their spouse, if they don't have longevity in the family, who share their environment with them. Okay, kind of share. You know, as somebody told me, I eat exactly like my wife, but twice as much. Okay, but you know, pretty good sharing the environment. They're also unique in the sense that they're Ashkenazi Jewish. This is just a genetic homogeneity because that's very important for my study. They happen to be also very educated, so I don't have diversity in things that matter for aging. But anyhow, 500 of the people that we tested are the opus, offspring of parents with usual survival, versus opal, which is offspring of parents with exceptional longevity. Okay, so all I want you to notice is I'm putting the opal ones, and what you just see with your eyes, that it's much more pink than the red on the left, right? It's much more pink, and the reason it's more pink is that in the opus, 585 are significantly changed, and in the opal, only 235 are significantly changed. In fact, it means that the opal, that are the same age as the opus, their aging is delayed by 8 to 10 years. Maybe in 10 years, we'll get exactly the same results with them. Now, that's not suggesting that, I didn't show by that they'll react to treatment, but at least on the genetic background, you can distinguish between them. Third point, sex differences. So important, we've the biologists missed it for so long. The ITP helped us know that when you give drugs, the longevity drugs to animals, the male and females can behave totally different. But this is the case with proteomic. Look, the yellow there is how many of the proteins were in females. Basically, you know, less than half the proteins. So female, when they age, have some of their own proteins, but in common, they have only part of the male protein. And one explanation goes to Morten's talk and to Alex's question, they're aging later, okay? So maybe just as I showed you that the Opel age later, maybe that's the case also with female. Maybe they're just younger, that's why we have less proteome. But when we develop a proteomic as a clock, we just have to know either we develop the proteins that are common to all of them, or we have some age adjustment or adjustment for sex at least for those proteome. The fourth important point, and that's actually really important, that's Paul and Laura, this is for you to, I'd like your thought about it, but basically, we have the concept that those SASP proteins that are secreting from senescent cells are contributing a lot to the plasma, okay? That some of those inflammatory markers that we're doing are really because of those things. Now, we collaborated with Judy Campisi, who's trying to get by different resources and experiments what's common to all senescent cells in the body from every organ and by many ways. And at that point, she found 174 SASPs, those are the secretory proteins, and we had 114 of them that we could measure, okay? And actually, out of them, 27% have changed significantly with aging. But let me show what happened, and this is so important. Basically, half of them went up, and half went down. Now, remember, those are all proteins that in our experiment were secreted, okay? I mean, I understand that some of the proteins will stay around the tissue and it wouldn't go up, but why is it going down? Okay, so I think this is not a proof, but this is an indication that the SASP are not really the majority that controls what we see in the plasma from this perspective. Okay. So how do we make, I showed you kind of raw data almost, how do we make clocks out of them? So some of you already, Alexei talked about it a little bit before. I don't want to go to how do you make clock, but you understand that the process is you have the proteins, you do it on a sample, you're training the samples, you validate on other samples. There's a whole process in that, and yet then you get clock. And this is the clock that we got from our subject that suggests the correlation between biological age and chronological age. So, you know, we have people who are biologically younger than their age, or in red, those that are biologically older than their age. In fact, this guy in red was 74 years old, but his predicted age was 86 years old. And he had high frailty, or she had high frailty, anemia, cardiac irregularity, diabetes, hypertension, gait velocity, BMI was 33, and she died 2.9 years after this. Okay, so there's some importance for the biological age, but of course, what we're trying is we're trying to do better. We try to do the age response to the number of years we have to live, or what's the age of death. And we have too little deaths in our subjects. Only 148 of our 2,000 people died. But you can see that we can get a very good and not such a different clock for mortality. We can also develop clocks for frailty, and we can show the relation. Okay, so sorry, something new. So for us, because we're looking at groups of people with exceptional longevity, who are resilient for genetic reasons and others, what we are trying to develop is scores for resiliency. So how do we develop frailty resilience score? We can go and look at the genetics that predicts frailty. And our question will be, those people whose genetic predicts frailty, how many of them don't have frailty, okay? That's how we are developing this in a way that the frailty resistance, okay, is associated with a measure of age acceleration. But the idea is that when we're talking with exceptional population, we have to define them as the resiliency, because this will help us. You know, so people who have all this frailty score but are resilient, why are they resilient? What are in them the genes that allow them to be resilient? And I think this is a concept that we start talking about, and I would like to develop it some more. Okay, it's 20 minutes, so I'm ending now. First of all, that you know, we have American Federation of Aging Research has the FAST initiative that is led by Dan Belsky and myself, but include a big consortium. And the idea is there's already studies that were done on a lot of the gerotherapeutics, on metformin, on lifestyle, on SGLT, on GLP-1. We're going to the sources that have samples of baseline and a year after, and we're going to omic the hell out of them, okay? We're doing everything in order to get a head start. Now, there'll be specific changes by treatment, but there'll also be commonality by treatment, and I think we'll get a head charge about that. So, just you know that's coming. So, I haven't demonstrated that the biomarkers are changing within weeks and months, okay? Or that proteomic is. I do think, though, that proteomic change rapidly. We already know, and that's Horvath and Morgan will tell you, most of the epigenetics is not changing so fast, not in a year. But also the biomarkers may provide insights for organ and disease-specific and weaknesses that we might have. So again, Morten and guys, thank you very much.
Thank you so much, Nir, for a fantastic talk. Always a pleasure to hear you speak. Questions for Nir?
Thank you for the great talk. Just a basic question. You mentioned differences between male and females. Which of these genes are X correlated and B through to X inactivation? Have you taken into consideration the X inactivation of these proteins?
Which mechanism are activating this protein?
What could be an explanation of the fact that you have X inactivation? It could be the dose is dependent. These proteins that they change between male and females?
No, I don't want to jump to mechanism. You know, one of the problems is with all those proteins, what's the approach? How do you cluster them? How do you find a common mechanism? And if anybody wants to collaborate on that, we have a core that supplies that.
All right, thank you so much, Nir. We have to end here because we have a short break now. Thank you so much.